The overall goal of this procedure is to detect active RhoC GTPase in cells. This is accomplished by first producing fresh GST-Rho binding domain protein. The second step of the procedure is to combine the GST-RBD with glutathione-separase beads. As the third step of the procedure, cell lysate is harvested and combined with the GST-RBD glutathione-separase complex in order to specifically bind active RhoC GTPase. The final step of the procedure is to precipitate out active RhoC, separate by SDS-Page, and immunoblot. Ultimately, results can be obtained that show levels of active and total RhoC GTPase through immunoblotting.
Hi, I'm Kenneth Van Golen from the Laboratory of Cytoskeletal Physiology in the Department of Biological Sciences in the Center for Translational Cancer Research at the University of Delaware.
And I'm Michelle Lucey, also from the Van Golen Lab.
I'm Heather Unger from the Van Golen Lab.
And I'm Erica Dashner, also from the Van Golen Lab.
Today we will show you a procedure for determining active levels of RhoC GTPase in mammalian cells.
We use this procedure in our laboratory to study the role of RhoC GTPase in cancer metastasis.
So let's get started. Let's!
Prior to the start of this procedure, prepare glycerol stalks of JM109 competent bacterial cells containing the PGEX3X vector encoding for the first N-terminal 90 amino acids of the rhotic Rho binding domain, or RBD, protein. For robust and accurate GTPase activation assays, fresh GST-RBD fusion protein must be prepared for each experiment. To begin, add approximately 50 microliters of frozen glycerol stock to 50 milliliters of Luria bertani, or LB, media containing